Hi, my name is Manasvi, and my mentor's name is Matthew, and my topic is the evolution of genetic engineering in medicine, especially focused in on recombinant DNA and CRISPR gene editing technologies. I chose this topic because I was interested in the field of technology in medical use. Currently, I'm presenting from Bay Area, California. So, to begin, genetic engineering and medical biotechnology are rapidly evolving fields which hold immense promise for revolutionizing healthcare through advancements established in findings related to recombinant DNA technology as well as CRISPR gene editing. The two main focuses of my pitch include recombinant technologies, which allow us to use aspects of other organisms' DNAs, as well as gene editing, which allows us to make edits, deletions, and substitutions in sequences. So let's begin with recombinant DNA technology. So this is the base of medical biotechnology, which sparked a path for a lot of development in this field. It was developed in the 1970s by Paul Berg, Stanley Cohen, and Herbert Boyer at Stanford University. It allows us to manipulate DNA sequences across different organisms, and it involves isolating specific genes or DNA parts from one organism and inserting them into another organism's DNA using either plasmids or strands of DNA. We usually tend to use E. coli or yeast or any other bacteria. To explain how this process works, over here we have the desired DNA fragment or trait, and it's amplified from a donor species using PCR, and restriction enzymes or other techniques are used to assemble these fragments. Replication occurs inside a host species, commonly yeast or E. coli. The recombinant DNA is introduced into the host organism where it's replicated and produces the desired trait or protein, such as insulin. So another thing to know about recombinant DNA is PCR, which I previously mentioned. PCR stands for polymerase chain reaction. It was invented by Mary Mullis in 1983, and what it basically does is make copies and amplifies the targeted fragment over and over again. So to show how this works, some of the basic steps are first, the double-stranded helix DNA strand is untwisted and separates due to high temperature, and the hydrogen bonds are broken between the two strands, and it gives us two separate ones. And then annealing is lowering the temperature, which then allows the short DNA primers to bind and start to create these new fragments attached to it. And extension is DNA polymerase creates complementary DNA strands, which then leads to two new double-stranded DNA strands, which are double the original amount. So it has two of the parental strands and two of the new strands. And this is basically done over and over again, and that's how it makes a bunch of copies of the same thing. Now let's talk about CRISPR-Cas9, which is a revolutionary gene editing tool. It was inspired by bacterial immune systems found in real life observations, and CRISPR Cas9 is a technology which was based off of that, and it allows us to make more precise and targeted modifications in genes. So, to target specific genes, scientists can design a short RNA sequence complementary to the specific DNA sequence they want to edit. This RNA sequence, along with the Cas9 protein, form a complex that can be recognized and binded to by the target DNA sequence. This holds promise for treating genetic disorders like sickle cell anemia or even Huntington's disease. So to explain how this works, over here, the guide RNA guides the Cas9 protein where to cut, and then the cell itself repairs the cut in a way that leads to a change in the DNA. The Cas9 proteins act sort of like a molecular scissors in a way, and it can make edits efficiently as well as pretty directly. This brings us to the ethical implications that are surrounding this debate on genetic engineering. Issues of safety, consent, accessibility, and long term impacts have to be considered in this. Pleiotropic effects and off target editing could lead to unintended consequences like mutations or cancer. Although there are many positives to this technology, it hasn't been tested long term in humans, and we don't know what the effects could be. While genetic engineering has many transformative and possible medical solutions, we have to take into consideration the ethical view. Some other additional factors to consider are who gets to use this technology and for what reason specifically, as well as making sure that if one certain group gets it, it should be accessible to all. So, in conclusion, advancements in genetic engineering hold promise for addressing medical challenges, improving human health in ways that we've never seen before, as well as recombinant DNA technology and CRISPR gene editing are significant milestones in medical biotechnology. Thank you.